Hi everyone, now we're on chapter 3 and this is a discussion about cells. This should be review for you because this unit was covered in your fundamentals of biology class that is required before you come into anatomy and physiology. However, this is very important to understand cell function, so I'm going to go over chapter 3 as a review. The cell is a structural and functional unit of life, and an organism's functions depend on individual and collective cell functions. The activities of cells are connected to their shapes and their subcellular structures. If you'll remember, these subcellular structures are typically called cellular organelles. The continuity of life is based on your cell's functioning. Cell diversity. In the human body, we have literally trillions of living cells, but there are over 200 different types of cells. Figure 3.1 is an illustration of some of the diverse shapes, and this will also lead to diverse functioning of various types of cells in the body. This part is showing you cells that connect body parts, form linings, or transport gases. Epithelial cells, you can see fibroblasts, erythrocytes, which are red blood cells. They carry oxygen to your tissues. Then here we have B. These are cells that move organs and body parts, skeletal muscle, smooth muscle cells. This is a cell that stores nutrients, a fat cell, or an adipocyte. This type of cell fights disease. It's called a macrophage. Here you see a cell that gathers information and controls body functions, a nerve cell or a neuron. And here is the human sperm cell. The generalized cell includes 3 basic parts: the plasma membrane, the cytoplasm, and the nucleus. And we will go through each of these. The generalized structure of a cell is represented here. There's not really a cell in the human body that looks like this, but this illustration here is just to show you all the various types of cellular organelles. Here's the nucleus, for example, the nucleolus, endoplasmic reticulum. Here are some mitochondria, the Golgi apparatus, etc. And we'll be going through these parts as a review near the end of the chapter. The plasma membrane. This is actually, if you'll remember, a lipid bilayer. It's actually called a phospholipid bilayer because here's the little phosphate heads with their lipid tails on the outside surface of the cell membrane by the extracellular fluid. Here is the phosphate head and lipid tails of the layer that is oriented toward the inside of the cell, towards what we call the cytoplasm. So this plasma membrane separates the intracellular fluid from the extracellular fluid. It's also, in reality, it has many structures that are embedded in it, proteins, various types of proteins. And scientists, when they were studying the cell membrane, they discovered that these proteins actually will migrate, move around a little along the membrane. And so the name fluid mosaic came from the fact that the proteins somewhat move within the membrane, and it looks like a mosaic pattern. If you've ever seen an artwork that is a mosaic, it has various colored glass or other materials forming a pattern. The plasma membrane is more accurately represented with this figure, and you can see all the various types of structures, proteins that are embedded and attached to the surface of the plasma membrane. Here's your little phospholipid bilayers, one here, one there. You can see some glycolipids. You can see a glycoprotein here, and all kinds of things we'll be talking about in a minute. So basically, there are a couple categories of membrane proteins. There are integral proteins. These include all the proteins that are inserted into the membrane. Their functions include transport proteins, either channels or carriers, some are enzymes, and others are receptors. And then there are peripheral proteins. Peripheral proteins are loosely attached to these integral proteins. Some of the functions of these types of proteins would be some function as enzymes. Others are motor proteins for shape change during cell division. Others, and particularly in muscle cells, perform muscle contraction. Others will form cell-to-cell connections. Overall, the 6 functions of membrane proteins will be, and each of these will be discussed individually, would be transport, receptors for signal transduction, attachments, enzymatic activity, intercellular joining, and cell-cell recognition proteins. The first one, transport proteins, is represented right here. So in this figure up here, you see 2 types of transport proteins. Some transport proteins over on the right, those on the left, that's discussed first. These are, they go all the way through the membrane, and they provide a hydrophilic channel, meaning it's filled with water. So things that are polar, they can't go through the lipids, so they can go through this channel. There's actually a couple of types of channel proteins, but this is passive, so that the substances will move down their concentration gradient from one side of the membrane to the other through these channel proteins. The one on the right here, this is a transport protein that hydrolyzes ATP as an energy source. In other words, this requires energy. It's called active transport, and it can move something from an area of low concentration to high, going uphill, up the concentration gradient, which requires energy. This would be a pump protein. Other proteins are receptors for signal transduction. So what's signal transduction? This is when a membrane protein that's exposed to the outside of the cell, it would have a binding site that fits the shape of a specific chemical messenger, such as a hormone. When this is bound, the chemical messenger is bound to that protein, it will actually cause the protein to change shape, and then that will initiate chemical reactions inside the cell, basically transducing a signal from receiving this messenger that binds to this protein, and then will transfer that information to the inside of the cell that will cause changes inside the cell. Other proteins will form attachment to the cytoskeleton and the extracellular matrix. Remember, extracellular means outside the cell. So elements of the cytoskeleton, which are the cell's internal supports, and then the extracellular matrix, which are fibers and other substances outside the cell, may anchor to these membrane proteins. And this helps maintain cell shape, and it can fix the location of certain membrane proteins. Others will play a role, other proteins such as this one will play a role in cell movement or bind adjacent cells together. Adjacent means those cells that are right next to it. Other membrane proteins will perform enzymatic activity. So a membrane protein may actually be an enzyme that will catalyze a certain reaction. So it will have an active site that's exposed to substances in the adjacent solution, right here. And there will be a team of several enzymes in a membrane that may catalyze sequential steps of a metabolic pathway. Enzymatic activity is often done in what we call a metabolic pathway, where one enzyme will take a substrate and will change it into a product, and then that will be passed along to the next enzyme, and it will take that enzyme's product, the first enzyme's product, and then it will change it and come up with another product. So this will happen along the cell membrane. I think of it somewhat like an assembly line in a factory where there's various workers, which would be they would represent the enzymes, and as they work on these, building a certain final product, there will be many steps along the pathway where this particular worker would take the raw materials, change it into one product, and then this worker would take that product and actually work on it and do another step and then pass it along. It will either be the final product or be passed along to the next worker. Other membrane proteins are going to form intercellular joining. Membrane proteins of adjacent cells, those are next to each other, might be hooked together to form intercellular junctions. And there's actually two types of intercellular junctions. One of them is called a tight junction. This forms an impermeable junction between cells to prevent molecules from passing through the intercellular space. This is showing an example of cells. These happen to be epithelial cells, for example, lining the small intestines, and they actually, epithelial cells typically are very close to one another. When they're forming a sheet, a tissue here, they will be tightly bound together with these tight junction proteins, and all these cells here you can see are attached to what we call a basement membrane to give them strength, but it's a sheet of cells that are attached tightly together. Different type of transmembrane protein would form what we call a gap junction. A gap junction is a little pore between the cells that would allow small molecules to pass from cell to cell. And here we see gap junctions right here where they're communicating junctions, and they allow small ions or ions and small molecules to pass between the cells. That would allow intercellular communication, so between this cell and this cell. Examples would be ions, simple sugars, and other small molecules that will pass, particularly between cardiac muscle. We have this happen where the ions will flow from cell to cell, and that would allow a nerve impulse to pass from cell to cell, so that the heart muscle will contract. Each chamber will contract as a unit. This also occurs with smooth muscle cells, and we will get to muscles later on in the semester. Cell-cell recognition proteins. These are actually called glycoproteins, as they have a little sugar chain attached to the protein. And these serve as identity tags. So it's like wearing your name tag when you're in an institution where the people that are that work there will know when they see your identification tag that you belong there. And so this is similar to the cell-cell recognition proteins in your body. If you think about your body containing trillions of living cells, how does your immune system know that those cells are your cells and not some foreign cell that has invaded the body? So these are very important, and so typically your immune system is trained to recognize your own cells as self. And so therefore, if there is a foreign cell in the body, the immune system will recognize it and get rid of it because it can be a threat to your life. So the plasma or cell membrane divides the inside of the cell from the outside of the cell. Cells are surrounded by interstitial fluid. This is the fluid between and out the cells, around the cells. So this fluid actually contains thousands of substances, and that plasma membrane will allow the cell to obtain exactly what it needs, but it keeps out what the cell does not need. Plasma membranes are therefore selectively permeable, or we can also call this semi-permeable. Some things can pass in and out, other things cannot. So there's actually two major ways that substances will cross the membrane. Some of the processes are passive, others are active and require energy. Passive processes include those that do not require energy, and of course cellular energy is provided by adenosine triphosphate, ATP. So passive processes don't need ATP. The substance will move down its concentration gradient from an area of high concentration to low. This does not require energy. For active processes, energy is required. Passive processes include diffusion, simple diffusion, carrier and channel mediated or facilitated diffusion, and osmosis. All three of these are types of diffusion. And then another type of passive transport, and this particularly is for water, it's called filtration. Filtration can occur across capillary walls. Your capillary walls are only one cell thick, very thin, and particularly if there's hydrostatic pressure, like in your blood pressure basically has a little water pressure behind it. So this can cause some water to actually filter out into the tissues, especially if the capillaries are, become more permeable to water, like during inflammation, etc. We will be talking about that a little bit later when we get to the lymphatic system. And also in the kidneys, there's a specialized capillary called the glomerulus, and that's where filtration occurs from the bloodstream during the cleansing of the blood. And of course, we have a whole unit on urinary system later. So, small molecules and ions are always going to move down their concentration gradients. The speed with which that happens is going to be influenced by particle size and temperature. The solute particle will passively diffuse through the membrane under three conditions: if it's lipid soluble, if it's small enough to pass through a membrane channel that is in the membrane, or if it's assisted by a carrier molecule. Lipid-soluble substances can diffuse directly through that phospholipid bilayer. So remember, fat and water do not mix. Things that are water-soluble may not get through that lipid bilayer, phospholipid bilayer. However, really small, non-polar molecules such as oxygen gas, O2, and carbon dioxide can pass through that phospholipid bilayer, and so can fat-soluble vitamins. So fat-soluble substances can go right through that phospholipid bilayer. And this is showing the simple diffusion of fat-soluble molecules directly through the phospholipid bilayer. Non-lipid soluble and polar molecules must be transported through some type of a protein carrier. So they will go from an area of high concentration to low passively, but they're going to be carried by protein carriers, or they can move through a water-filled channel. So examples of these substances would be amino acids, glucose, and various ions, charged particles. Figure 3.7b shows this diffusion through carrier-mediated facilitated diffusion. So you can see here the substance is in greater concentration on this side of the membrane than down here, so it will go passively, but it needs to be carried through some type of a carrier. It needs to be transported using this channel because it's lipid insoluble, things like sugars or amino acids. So this carrier protein will bind the substrate here, we call this a substrate, and then it will transport it to the other side of the cell membrane. Channel-mediated facilitated diffusion. A channel is just what it sounds like. It's a little passageway. There are actually aqueous or watery channels that are formed by transmembrane proteins. There's two major types of channels. There's leakage channels that are always open, And then there are gated channels, and these have a gate. When the gate's open, they can freely pass through. When the gate is closed, they cannot go through. This figure is showing channel-mediated facilitated diffusion through a channel protein. This is mostly going to be ions. Of course, remember, ions have a charge to them, either positively charged or negatively charged. And these channels are very specific for each type of ion. So there are sodium ions. So there are chloride ions. There are sodium channels for sodium ions. There are potassium channels for potassium ions. There are chloride channels for chloride ions. Some of these channels have the gates, which will open and close. Other ones do not. So they're just representing both right here. You can see this passageway, which is a channel. Osmosis. Osmosis is really the diffusion of water across a selectively permeable membrane. This is somewhat of a review. You should have encountered the concept of osmosis during your fundamentals of biology class, but just as a little review, water will pass through plasma membranes through the lipid bilayer in very small amounts, but mostly the water will pass through water channels that are called aquaporins. Water will diffuse down its concentration gradient to the side in which there are more solutes. So I like to remember this phrase. It goes to the side in which there are more solutes. This is showing right here you have an aquaporin. These are those little water molecules, and they will pass through this channel. It's called an aquaporin that will allow them to go through much more quickly from one side of the membrane to the other through these aquaporins. Solute particles, remember, displace water molecules. The definition of the term osmolarity refers to the concentration of solute particles in a certain solution. Osmosis will occur until hydrostatic pressure and osmotic pressure equalize. Hydrostatic pressure is water pressure. So if you are filling up a balloon with water, as you keep filling that balloon with water, the hydrostatic pressure is stretching it so much, eventually that balloon will burst. Water pressure is quite heavy, actually. Water pressure is significant. That's called hydrostatic pressure. And then there is osmotic pressure, which we're going to look at these. This is showing you a membrane that, right here, here's the freely permeable membrane to both solutes and water. So if you separate these two areas with a permeable membrane, here you have the amount of water is equal, you've got fewer solutes over here, and you have more solutes over on the right. Eventually, these will all equalize. They will go to equilibrium because both solutions now have the same osmolarity and the volume of the water will not change. However, if you have a membrane that is permeable to water but impermeable to solutes, and this is true in the human body, particularly with larger solute particles. Okay, so we have the left compartment and the right compartment. This is a selectively permeable membrane, which is what cell membranes are. And so you can see over here there's fewer solutes. Over here there are many solute molecules. This would be a sugar that cannot pass through that membrane. So what's going to happen? Water will diffuse. Water actually can diffuse back and forth, but there will be a net diffusion from this area over to this area because the number of solutes over in the right compartment was much greater. So the water will be drawn over to that area where there are more solutes, and therefore it's going to increase the volume now on the right side. This is really important in the body because osmosis will cause living cells to swell or shrink depending on what type of fluid that they are surrounded with. A change in cell volume either way will disrupt cell function, especially in neurons. There's a few terms here that are associated with osmosis, and it's called tonicity. Isotonic, and we're going to show these in the next figure, isotonic, hypertonic, and hypotonic. So here is a figure 3.9, and it's the effect of solutions of varying tonicities on living red blood cells. So this would be when you infuse a solution directly into someone's bloodstream. If you are using an isotonic solution, that means it has the same relative number of solutes in it as that in normal blood. And keep in mind, the surrounding fluid around red blood cells in your blood is going to be the same as the fluid inside of the cells. And this is about 0.9% tonicity. So you want to infuse something that is isotonic, like normal saline, which is 0.9% sodium chloride. When you infuse this fluid, it's going to be the same as the tonicity or the concentration of solutes that is inside the red blood cells. So the water can go in and out from the cell, but there will be no net flow either into the cell or out of the cell. Now let's just say that we put a hypertonic solution into the bloodstream. This is a solution that has a greater tonicity, or in other words, a greater percentage of solutes in it. And so the cell will actually lose water. There will be a net flow of water from inside the cell out to the fluid that has a much greater number of solutes in it, and therefore the cell will shrink. If you infuse a hypotonic solution, let's just say sterile distilled water, which has virtually no solutes in it. Okay, so now you're surrounding this red blood cell with a hypotonic solution that has fewer solutes than the fluid that's inside of this cell, which remember is about 0.9% sodium chloride, just as normal blood would have surrounding that cell. But you put this hypotonic solution, distilled water, into the bloodstream. It will surround these cells, and because there's a greater concentration of water outside now, it will actually be forced into the cell. Because remember, the cell has a greater number of solutes in it. It is a 0.9% sodium chloride, plus there's other suspended, particles in here. So the water is actually going to be sucked into the cell, and the cell will burst. Table 3.1 is a review of passive membrane transport processes, and that is the end of the show. And so have a wonderful day. I will see you with the next one, which will be Chapter 3, Part B.